This video will instruct you on how to prepare the solution containing your biotinylated material with Streptavidin ZAP. This part of the assay will be performed under sterile conditions, so the next steps will be done in a cell culture hood. First, assemble the materials you will need: a well stand, which is optional, pipettes with appropriate pipette tips, a well reservoir, the appropriate cell media for your cells, your biotinylated material, microcentrifuge tubes, the plates of the cells you plated the previous afternoon, and of course the Biotin-Z Internalization Kit. Please refer to the PDF file on the flash drive included with the kit for complete written protocol instructions. Begin by opening the kit and removing the Streptavidin ZAP and setting that aside. Then remove the well template. You will notice that columns 2 and 11 are marked for the controls. Place the template on the well stand. The first step is to transfer 10 to 15 milliliters of cell media to the well reservoir. Then, you will label the microcentrifuge tubes with the numbers 1 through 8. The calculation worksheet to determine the proper ratio of Streptavidin ZAP to your biotinylated material is included in the written protocol instructions and is also available on our website. To tube number 1, add the calculated volume of biotinylated material. Then, add the calculated volume of Streptavidin ZAP to tube number 1. Pipette up and down to mix and then vortex briefly. Let the material incubate at room temperature for 15 minutes. During the 15-minute incubation, prepare the solution of Streptavidin ZAP spiked media only. Take a microcentrifuge tube and label the cap ZAP only. Then, to this tube, add the same volume of Streptavidin ZAP that was calculated for tube number 1. Next, add cell media to this tube calculated to be 150 microliters minus this volume of Streptavidin ZAP. Vortex briefly afterwards. After the 15-minute incubation, transfer 135 microliters of cell media into tubes 2 through 8. Next, we will perform the serial dilutions. Taking tube number 1, add cell media to bring the final volume to 150 microliters. Pipette up and down and vortex briefly. Begin the dilutions by transferring 15 microliters from tube number 1 and transferring it into tube number 2. Vortex briefly, and repeat by transferring 15 microliters from tube 2 into tube number 3. Continue to repeat these steps through tube number 8. Once you've finished making your dilutions, you are now ready to begin adding the material to your plates. Begin by placing the well template underneath your plate. The first step is to add to the control columns. You will add 10 microliters of cell media only to each of the 6 wells, B through G, of the control column number 2. That means the first and last wells in the column will not receive media. Then, you will add 10 microliters of Streptavidin ZAP spiked media only to each of the 6 wells to the control column number 11. Then, starting with tube number 8, vortex briefly and transfer 10 microliters into each of the 6 wells to the column labeled tube number 8 according to the template. Repeat this step for each of the dilutions. Tube number 7 into column labeled tube number 7, tube number 6 into column labeled tube 6, and so on until tube number 1 into the column labeled number 1. Once you complete adding the material to the plate, place it back under the appropriate growth conditions for your cells for 72 hours.